introduce Dr. Sean Ron Keith Kelly, Associate Professor of Surgical Oncology, Endowed Professor Ronald E. and Patricia A. Kissinger Surgical Oncology Research Professorship. Uh, Sean did his medical school at University of Colorado, a general surgery residency here at the University of Wisconsin, and a complex general surgical oncology fellowship at Johns Hopkins. Sean is certified by the American Board of Surgical uh, as a general uh, uh, in general surgery and specializes in malignant disease and pancreatic liver. Sean's lab focuses on the formation and progression of pancreatic cancer, as well as the understanding and communication within the tumor microenvironment. So welcome, Sean.
Awesome. Thanks for the introduction, Derek. Can you all hear me okay? Great. Well, thanks for the opportunity to speak today. I was going to be talking about some of our work in fibrolamellar carcinoma, or more specifically, PRKCA fusion cancers. So these are comprised of pancreatic ductal adenocarcinoma and cholangiocarcinoma, or cancer of the bile duct. And as you all probably know, these are bad cancers, poor survival, few treatment options. And so the specific subtypes of pancreas and cholangiocarcinoma are actually called intraductal oncocytic papillary neoplasm, or IOPN, associated carcinoma. And basically what that means is that's just the precursor lesion that leads into the cancer. That's what they're classifying there. And most pancreas cancers and many of the bile duct cancers have a KRAS mutation, but that's not the case for these IOPN-associated carcinomas. They're actually driven by these PRKCA fusions. So ATP1B1 PRKCA and the prototypical DNA JB1 PRKCA. And so this fusion was actually discovered roughly 11 years ago in fibrolamellar carcinoma. And that was very fortuitous because this is another cancer where it's poor survival. So only 40% survive after 5 years of diagnosis. And again, a cancer with really no proven treatment options. And so you have this gene fusion that arises in the pancreatic duct or bile duct for these IOPN-associated carcinomas, and an unknown cell of origin for fibrolamellar carcinoma. It's in the liver, but we don't quite know what the cell is. And it leads to these deadly cancers. And so this is an MRI of a patient with fibrolamellar carcinoma. This is the liver, and you can see the tumor there. And this is particularly devastating because of the fact that fibrolamellar carcinoma is diagnosed at an average age of 21 years. And so if you have a 40% 5-year survival at age 21 years, that's pretty bad. So we have a gene fusion here that actually causes, or is thought to cause, 3 separate types of cancer. Most is known about fibrolamellar, but the IOPN-associated carcinomas have actually just come to light. There isn't much known about them. And so our assertion is that these IOPN-associated carcinomas and fibrolamellar carcinoma, or FLC, have the same pathology or pathobiology. The reason why this is important is that when we discover therapy for one cancer, we can now apply it to all 3. And so to begin to investigate, we developed the mouse model. This was in conjunction with the Genome Editing and Animal Models Core, Dustin and Kathy. And so we started with an AI14 marker mouse. And so this is just a conditional allele of the TD tomato red fluorescent protein at the ROSA 26 locus. We call this the AI14 marker mouse. So when you cross it to a Cree expressing animal, wherever Cree is expressed, you remove the lock stop and you get red fluorescent protein in that cell type. We used CRISPR-Cas9 to introduce the gene fusion followed by a linker in front of the TD tomato, and this allowed us to generate our target mouse model, as you can see here. We then crossed this to cytokeratin 19 Cree ERT mice, and we specifically chose this because cytokeratin 19 is expressed in the bile duct. And so this then allowed us to sort of specify the cell type where the oncogene is being activated. And by having the Cree fuse to a modified estrogen receptor, we could also control the timing of onset of the allele activation. And so we call this the AI14DP KRT19 Cree ERT mouse. And so when we cross these mice and we administer tamoxifen, within 3 weeks, we start to see these lesions that form. And so these cells are characteristic cells that we see in human fibrolamellar cancer and human IOPN-associated carcinomas. They have enlarged cytoplasm, enlarged nuclei. The cytoplasm is granular, and it's a reflection, and we call it eosinophilic or oncocytic, and it's a reflection of densely packed mitochondria, which is very characteristic of these cell types in these tumors. When we allow these to progress longer, they move into these advanced eosinophilic proliferations, and these are consistently identified around the bile duct, which is what you can see here in the circle, because of the fact that they're arising from the KRT19 expressing cells. And of note, previous models where they've tried to generate mice that harbor this DP fusion, this is the extent of the tumors that they have been able to achieve. And so this is neat to see, but it's not the full sort of spectrum of what we would expect in terms of these DP fusions. So what we actually see as we allow these mice to age more in our model is early intraductal oncocytic papillary neoplasms, or IOPNs. So we can see the sort of characteristic papillary projections and cystic lesion here. They're all lighting up red, indicating that all those cells harbor the gene fusion. These progress into advanced intraductal oncocytic papillary neoplasms. And when we line this up from a specimen from a patient, we can see here that there's oh, thanks. So when we line this up, great, got a pointer now. We can see here that there's a pretty nice matchup in terms of the histopathologic features between our mouse model and human IOPN. And specifically, we're looking at some of these salient features such as arborizing papilla, neoplastic cells with abundant eosinophilic cytoplasm, granular cytoplasm, large nuclei. And so the papilla that they're talking about is basically these papillary projections that are sticking out here. And these are the cells that are enlarged and eosinophilic. They're also the cells that harbor TD tomato, indicating the DP fusion. The other thing that's important is that our fusion model develops this characteristic fibrosis. So on the right is a human sample, on the left is our sample, or our mouse model. And fibrosis is a defining feature of all three of these tumors in humans. And so if you don't have the fibrosis, there's something wrong. And this is the problem with some of the previous mouse models, is none of them developed fibrosis, and so it didn't actually recapitulate the tumor microenvironment, which is a critical factor when you're trying to evaluate tumor onset, response to therapy, et cetera. The other thing that was very exciting to see is that our mice have tumors that progress to cancer. So this is cholangiocarcinoma. And basically what you see is the sort of IOPN precursor here, but here we have obliteration of those papillary projection cystic areas. And these are all cancer cells lighting up positive, TD tomato positive. When we take whole liver from these mice, so this is inclusive of tumor and adjacent normal, and we stain or assess for PKA, native PKA, using an anti-PKA antibody. In the controls and the tamoxifen-treated mice, we see native PKA, and then only in our tamoxifen-treated mice with the activated allele do we see the oncoprotein. And so DP has exons 2 through 10 of native PKA, and so to pick this up, you simply use anti-PKA, which gives you native and DP. This is what we see in humans when we're evaluating at the protein level. We also assess for sort of pathognomonic gene expression. And so there are a set of genes in fibrolamellar cancer that are consistently upregulated, and these are super enhancer associated genes, or we call them FLC specific genes. Two of them are depicted here. We see that these are markedly upregulated in our whole liver compared to the corneal treated and C57 controls. And then finally, when we look at survival, we can see that all of the control mice survive 100% to the study endpoint, whereas our study mice that have the tumors only survive out to about 3 months, which is actually a huge benefit because it means we have more rapid tumor onset, which allows us to study the pathology a little bit easier than if it was a year or 2 years to develop tumors. Additionally, we not only see tumor development in the liver, the same thing happens in the pancreas. So we see IOPNs in the pancreas, and then these progress to IOPN-associated pancreas cancer. Again, all lighting up red, TD tomato positive. These harbor the DP protein, and they also have FLC-specific gene expression upregulation. So this is critical because the IOPNs are parallel entities that occur in the bile duct and the pancreatic duct, and they don't occur elsewhere. So the fact that we're able to recapitulate these in both in one mouse model is pretty neat. So our data to date supports actually that these three cancers do harbor the same biology. So we see the same or identical pathologic features in large oncocytic cells, fibrosis, development of carcinoma. Additionally, we see presence of the fusion oncoprotein and FLC-specific gene expression. And so this is a critical advancement because it has significant implications for future work. Up until this point, we didn't really have a good model system in vivo to evaluate any of these cancers. Previous has been either patient-derived xenografts, but these are all very rare tumors. And so it's really hard to obtain tumor tissue or cell-based models, which have limitations. So here, with this model system, we're able to make significant advancements, including looking at early mechanism, looking at the tumor microenvironment, evaluating the immune microenvironment. And there's a lot of interest about this as well because the difference between a patient-derived xenograft model and this model, this is immunocompetent, and so we can actually evaluate the interaction with the immune system. So this is going to be a helpful model in addition to some of the other models that are out there for evaluating these PRKCA fusion-driven cancers. One of the other things is that we can implement therapeutic testing in an immune-competent model. And then perhaps of most interest is target identification because at present there are no proven systemic therapies for these cancers. One of which we have significant interest in is SLC16A14. So I was going to transition to talking about that for just a little bit. So SLC16A14 is a channel protein in the solute carrier family. And this was actually discovered in 2020 by a colleague and collaborator at Cornell. So they basically took human tumor samples and adjacent normal liver and used a technique called CRO-seq, where they're evaluating nascent RNA to identify enhancer hotspots or super enhancers. They then paired those up with adjacent genes to identify the super enhancer associated genes. And super enhancers are thought to be critical for cancer cell identity. So they drive high levels of expression of genes that are essential for cancer cell function and survival. And so through this, they were able to identify a set of genes that were dubbed the super enhancer associated genes, one of which was SLC16A14. And so this is markedly upregulated in tumor compared to normal tissue, somewhere around 2,000 to 3,000 fold, as we can see here. And this was followed up by a study in Sanford Simon's group. Simon was the one who discovered the gene fusion. And they also identified that SLC16A14, highlighted here in green, is markedly upregulated in tumors. So this is RNA sequencing data here, roughly 20,000 counts in the tumor compared to roughly 100 in the adjacent normal liver. So this is pretty exciting to see because of the fact that regardless of the patient data set, regardless of the lab performing the study, we see that this is markedly upregulated. So my colleague at Cornell took this a little bit further. He did single nucleus RNA sequencing study and looking at SLC16A14 expression across the various cell populations. We find that it's actually only upregulated in the epithelial cells that are thought to contain the gene fusion. as opposed to all the other cells distributed throughout the specimen, including the normal hepatocytes, as you can see here. What's more is that when we look at this SLC16A14 expression in FLC primary tumors and metastatic disease, it's markedly upregulated and essentially not expressed or minimally expressed across all normal tissues throughout the body. So this sort of reflects the conundrum with this protein. We don't really know anything about it because it's never come up before as being important. It doesn't seem to serve any critical function in normal cells. There isn't much data about it. We don't know what it transports. We don't know if it's a co-transporter. So there's a lot that's unknown, but what is key is that we don't find a lot of expression across the normal tissues. And then when we look across the different cancers, we actually see that it's only upregulated in PRKCA fusion-driven cancers as opposed to all the other cancers. And so this is really exciting because now we have a very unique target that is not well known, that's only upregulated in PRKCA fusion cancers, and is not expressed in other tissues throughout the body, which is probably the best target that you could possibly hope for. But I guess that is yet to be determined. So to investigate this further, we generated a cell-based model. We started with HepG2 cells. These are hepatoblastoma cells derived from a 16-year-old patient with liver cancer. And as a reminder, DNA JB1 and PRKCA sit on chromosome 19. They're approximately 400 kilobase pairs apart. So using guide RNA Cas9, we're able to achieve deletion and recombination to introduce the gene fusion at the endogenous locus. And so these are called HepG2 DNA JB1 PRKCA or HepG2 DP cells. We also call these the H33 and H12 clones. So these have a wild type SLC16A14 or two wild type alleles. And when we evaluate expression of SLC16A14 compared to the parent HepG2, we see marked upregulation as expected. We then took the H33 clones and we generated a knockout construct. So using CRISPR-Cas9, we knocked out 715 base pairs of exon 4. We chose this one because this was the most conserved exon across the isoforms. And so the SLC16A14 knockout version is what we call the SK clones. So we confirmed this with PCR genotyping. As we can see, the parent cells, the HepG2 DNA JB1 PRKCA clones, H12 and H33, they have wild type, and our SK only have the deletion construct that we can see. We then partnered with GeneCore again, who helped us with doing amplicon sequencing. This is actually pretty outstanding sort of service that they were able to provide for us because it really gave us more confidence in terms of what we were looking at. So part of the problem with generating these clones is sometimes we lose or gain various other gene-edited components. And so we first wanted to make sure that all of our clones had the fusion. So we tested the HepG2 DP cells. And this is basically across the breakpoint here. And so as you can see, we're getting reads across the breakpoint. So this is the combined up here. This is this region that's across this gene fusion here. And we get strong reads across indicating that this is in fact intact. And importantly, it's also intact in our SK clones. So we did not lose that gene fusion in the process of generating those clones. We then looked at the SLC16A14 exon 4 region. And as we could see in our HepG2 DP cells, we get great signal throughout this region here. And then when we look at our SK clones, all of them had this bottoming out or drop off where we had no signal, which confirmed for us that we did in fact have a deletion construct here. So we then tested for mRNA expression, massive decrease in mRNA expression. I'm actually not sure what's being picked up here. These are probably non-functional fragments. Unfortunately, none of the protein antibodies work all that well. So we have not been able to confirm this at the protein level. We're working on actually generating better antibodies. And so with the extent of validation that we were able to perform, we then moved on to investigate SLC16A14 function or try to get a glimpse of the function. So as I pointed out before, the cells in fibrolamellar cancer, IOPN-associated carcinoma, generate these large eosinophilic cells. And this is because these cells just become packed with mitochondria. And so there's a profound metabolic shift that occurs in these cells. And so the fact is that the SLC family is a family of solute carriers that are critical for metabolic functions. They transport things like lactate, pyruvate. They transport protons out of the cell. So to assess and see if SLC16A14 might be involved in metabolic function, we did sort of a global test to start with. We used the seahorse cell mito stress test. And basically the idea is that you measure oxygen consumption rate over time. You administer these various agents at various time points, which then give you a gauge of various mitochondrial functions such as basal respiration, ATP production, maximal respiration, et cetera. So looking at our HepG2 cells, so the parent cell line, we see a nice curve here. So this is sort of a great baseline to see. And then when you introduce the gene fusion, you see a massive increase across the board in terms of metabolic function. Interestingly, when you knock out SLC16A14, it essentially reverts almost back to baseline. This was actually kind of a surprising finding. I didn't expect it to be that profound. And so I can go through each of these step by step. But when you look at basal respiration, that goes up substantially with the introduction of the gene fusion, essentially reverses when you knock out SLC16A14. When you look at ATP production, that goes up threefold with introduction of the fusion. And again, you knock out the channel protein and that reverses. Same thing for maximum respiration, although not to quite the same degree, mitochondrial oxygen consumption and proton leak. And so when we had these findings, this convinced us, one, that we had sort of validated the cell lines, and we are going to be able to use these to evaluate SLC16A14 in these cells. And then two, this was a very interesting metabolic phenotype. And so this sort of gave us the courage to reach back out to Dustin and Kathy to generate our next step, which was the mouse model. So we wanted to generate two separate in vivo models. One of them was a global knockout mouse. And then for this line, we have the founders. These have been back crossed, and at present we're doing the allele auditing, and also the floxed version. So basically, we just used the same construct that we had in the cells, where we targeted exon 4, and then for the floxed version, the LOXP sites were basically just on either side of that 715 base pairs. So in partnership with the Gene Expression Center and Sequencing Core and GEME, we were able to do basically this validation or allele auditing, which is, in my opinion, pretty darn cool. Just because previously you would generate mice and you weren't quite sure, or you didn't have high degree of certainty. But basically providing a mouse, this was a heterozygous mouse, evaluating the SLC16A14 locus. And we did long read whole genome sequencing, and it was about 10x coverage using Oxford Nanopore. And as you can see from the different reads here, what we're interested in is whether or not these LOXP sites were integrated as expected. And when we look at these, we see about 10 reads here, and 6 of them harbor the LOXP sites, which is what you would expect in heterozygous, which is really awesome to see. The other great thing about the allele auditing is you can look for off-target impact. And so I think that this dramatically increases the confidence in terms of the mice that you're using and propagating, crossing to other models, basing all of your subsequent science on. So this was really, really nice. So at present, we have these mice, and we're crossing them to our IOPN-associated carcinoma model, with the idea being that we'll be able to evaluate phenotype differences, survival differences. We'll be able to use these mice for additional follow-up studies to investigate SLC16A14 as a target in PRKCA fusion cancers. So this is the AlphaFold sort of predicted model of this protein. This is the amino acid sequence here. It's predicted to have N and C terminus as both cytoplasmic. So some of the other family members in the MCT family have kind of a similar construct where it's a 12 transmembrane protein. N and C terminus are both cytoplasmic. And so this sort of follows that as well. Ben Minkoff ran this through the deep TM to sort of project what would be the transmembrane components. And so that's allowed us to glean a little bit of information. But currently, we're performing localization studies. We think this is a plasma membrane protein. All the other family members that have been evaluated are plasma membrane. We also are very interested in what this is transporting, because clearly there's something critical for the cells that SLC16A14 is transporting, or its function, and whether or not this is transporting in or out of the cell. We do have evidence, some evidence that it's likely a proton co-transporter. So we have a couple of different experiments that we've run where it seems that the extracellular acidification rate is going up. And that's consistent with the MCT family 1 through 4 proteins. Additionally, the key question is, why is this important for the DNA JB1 PRKs expressing cells? And so there's a lot of different things that we can investigate, which could lead us to potential therapeutic targets in these spectrum of deadly cancers. So that's essentially all I wanted to share today. Obviously, a huge thanks to everyone who's been involved in this, and the Biotech Center, who over a period of, I guess, 6 or 7 years now has really been instrumental in helping us to progress this work. So thanks all. I'd be happy to answer any questions. How do I get to the chat? Does anyone? Oh, Derek, you had a question? Oh, okay. Yeah, so that's, so the question was, how conserved is the protein across human mouse? So, that's a good question. In terms of the percent homology between human and mouse, I don't know the exact percentage. So recently, what we've actually looked more into is the percent homology between the different MCT family members. So there's 1 through 14 proteins, and we've just specifically been looking at humans. But there's not a significant amount of homology between the different family members. And so it's somewhere between 26 and 40%. And so there's a lot of conserved components that are like transmembrane components or N-terminal components. But yeah, that's something that will be important to think about as well as we're using our mouse model in comparison to human models. But good. Yeah, that's a good question. So that currently is unknown. So Sanford Simon recently published data, and sorry, I'll repeat the question. The question was, how do you envision how this gene fusion causes remodeling of the epigenetic landscape, emergence of super enhancers to drive expression of genes? So the native pKa is bound by pK regulatory subunits. And so basically with cyclic AMP, it releases pKa, which allows it to then phosphorylate its targets. It's preferentially sort of scaffolded in certain parts of the cell. And what Sanford Simon recently demonstrated was with addition of DNA-J onto the front aspect of the protein, it loses the regulatory function. So now you actually find it throughout the cytoplasm in places that you wouldn't necessarily find it with native pKa, but more importantly, also in the nucleus. so to a much greater degree than what you would see with native pKa. So what it does at that point to cause the remodeling, that's a good question. I don't quite know the answer to that. We have been looking at its impact on RNA polymerase II via CDK7, and it does seem to be that CDK7 is greatly activated in these DP expressing cells. And likewise, when you use CDK7 inhibitors, it's pretty lethal for these cells. But we don't know what the connection is specifically. It's basically, so to define a super enhancer, all they do is they basically graph the enhancer hotspots, and on your x-axis is basically the different enhancers, and then on your y-axis is sort of the levels of gene expression that they're associated with. And there's an inflection point where it starts to ramp up. And so you do like a tangent with the slope of 1, and then beyond that tangent at the inflection point, everything above that is considered the super enhancer. So it's actually just a statistically defined entity. And so that's how they come up with that. And so, you know, looking through the literature, when you're looking at what defines or what is a super enhancer, using that definition, they found that they preferentially cause expression of genes that are seemingly important for the function of the cell. And so that was sort of what was narrowed down, is like, hey, we have this group of genes that seem to be upregulated consistently. They're highly expressed, and they seem to be important for cell function. But to my knowledge, nobody's actually gone through and evaluated the specific function of any of these genes. It does live. Yeah. They're viable. We've been crossing them. So, yeah. 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 So, yeah, that is, I think that's going to be a challenge, obviously. The SLC family members have just started to come to light as therapeutic targets in cancer. And so I think preliminarily there's enthusiasm about generating small molecules towards those. But from what I've seen, there isn't anything that's generated as of yet. I think that part of the problem that I've seen in the literature is there's a lot of off-target effects with inhibiting these. And so let's say you use siRNA, well, you're probably targeting, you know, other family members. And so there is a lot of overlap. And then part of the other conundrum is that actually there's a lot of redundancy, like similar to the clock proteins. You actually have within a family 2 of them that when you knock one out, the other one picks it up. But if you knock them both out, now all of a sudden there's complete loss of function. So we don't know that yet with SLC16A14. But yeah, I mean, the idea would be that if we identify what the substrate is that's being transported, we could then generate a small molecule inhibitor that mimics that and essentially, you know, blocks that function. Correct. Yeah. And actually, you know, the knockout was critical because we did a lot of the inhibitor, you know, like the siRNA and things like that. We got great results in the DNA JB1 PRKCA expressing cells. Like you use the siRNA, RNA levels go down dramatically. The cell dose-dependent death curve is beautiful. And then we tested cells that don't have any expression of SLC16A14, did the exact same thing. And so I even looked back at our RNA sequencing results that we did with the Gene Expression Center. And I was looking at it, and the RNA levels of expression in the cell line that we tested was like 1, 2, like essentially negligible. And I was like, oh, well, I don't know what we're targeting then. And so, yeah, it really was, beneficial to generate the knockout versions. Dustin. Yeah. So yeah, the solute carrier family, they're definitely on the mitochondrial membrane or plasma membrane. And actually, some studies suggest that that can actually change depending on the environmental conditions, which is pretty interesting. Um, the MCT family, the monocarboxylate transporter family, of which SLC16A14 is a part of. So it's the SLC16A is MCTs. They have different nomenclature. They seem to be uniformly plasma membrane proteins. And so we do have some evidence that it is, in fact, a plasma membrane protein using GFP tag studies and confocal microscopy. But there's a lot of unanswered questions currently. So, Sandra, sure. Yeah, yeah.
Late 90s, cancer, and then he progressed to disease and died. And after he died, it's been called the kind of rare form of biochemistry.
Mm.
And since then, I've read different things that they are attributing that advancement in liver biochemistry to the pain medication that we want. That died,
That would be an amazing tumor if that was the case. And so it's probably that it arose from, that sounds more consistent with a bile duct cancer, just because of the aggressive nature. So typically, liver cancers are less aggressive if they're arising from the hepatocytes. It's not always the case. But cholangiocarcinomas in and of themselves are rare. And then there's a lot of different subtypes that are dependent on the driver gene mutation. And so, we're finding out a lot more about these cholangiocarcinomas. But yeah, I suspect that it was just probably all along a bile duct cancer that was just large and took up a substantial part of his liver. But I think that sort of speaks to the fact that a lot of times we try to treat these cancers the same. We say, oh, it's a bile duct cancer. We're going to give them this therapy. And that's what we have currently. But we're getting a lot more sophisticated now. And so we're finding these different, fusion genes and targets that allow us to be way more selective in what we're providing. But we're not quite there yet in terms of, first line therapies and things. Typically, those are reserved for second line or things like that. But yeah. That's a good question. So in our mice, we actually see the equivalent of sarcomas and or soft tissue tumors, and they also basically have a lot of these similar features. So it's possible, and that we just haven't identified them yet or subtyped them. Because the only reason why we know about them in humans is because we started looking for it. The only reason why we figured it out, or Sanford Simon figured it out in 2014, was because he was trying to investigate what was going on. Because there was no, in the fibrolamellar cancer, there were no other pathogenic drivers. There was no KRAS, there was no P53, there was no CDKN2A, nothing. And so I think part of the problem is we've only recently started to look for this. It's now part of a panel, like Foundation Medicine panel and things like that. And so hopefully like every tumor is being sequenced, and that way we can learn more. But it is possible that there are other tumor types. Dustin. That I don't know. I mean, I will be interested to evaluate the global knockout mice just to see, from a histology standpoint and such, to see if there's any differences. But sorry, what'd you say? Oh, I see what you're saying. You mean doing more of like a immunophenotyping or something like that? Yeah, that I don't know. I mean, I think part of the problem for us is going to be a balance of sort of direction and cost. And so how much are we going to be able to investigate that? But yeah, I mean, I think the good start is that they're viable and fertile. So that's helpful.
Second, these are patients scan fitting where the close to it. That close to that well then goes along body that or they're hitting old well.
Yeah, so there seem to be two peaks, and the main peak is, at the sort of late adolescent and early 20s. And so you have 15, 17, 19, 21, around there. And then there is a second peak that's a lot less common for fibrolamellar cancer, that is. And that's in like the 40 to 50 age range. The tough part is you analyze the livers, the normal livers, and there's no associated mutations there. This onset is a somatic event. They don't have any other, underlying susceptibility like a BRCA gene mutation. The livers otherwise appear healthy and normal. So there isn't much known about why these arise. But I think part of the challenge for doing any sort of association too is the rarity of it. So we don't yet know what induces this.
All happen, but don't happen.
Yeah. Yeah, that's actually a really good question. And we at least see in our mouse model that there are several areas that have the fusion that have these pre-neoplastic or precancerous lesions that don't develop. Like not all those spots are developing into cancer, at least in the time frame that we see. I don't really know. That is a good question. It would be interesting to try to figure that out. That has definitely not been investigated in humans. Um, and I don't know how closely we would have to scrutinize to be able to get to that ability. Yeah, that would be an expensive undertaking, I think. I think I think the tough part though is so you're also looking at um, where are you getting the tissue from, and you know I can't just come to your door and be like, Chris, I'm going to biopsy your liver because I'm looking for this mutation. Yeah. And the fact that there are different fusions sort of connecting the same region is also an indication that this seems to be a favored spot for generating the fusions. But yeah, I don't know how would be able to get to that though in patients from a ethical standpoint, that is. You you're saying in wild type mice? Mm. All of us actually probably have pancreas cancer precursor lesions in us right now, based on autopsy studies. So it is it is concerning. But yeah, that is the thing is that even if you have onset of these mutations, it has to acquire additional characteristics like immune cell evasion, et cetera, in order to progress. Thanks, everyone.